Hello, everyone. Welcome back to our beginning of our next installation of the Bodega Marine Laboratory Seminar Series. My name is Dr. Alyssa Griffin, and I'm very excited today to welcome and introduce Dr. Emily Bachman. Dr. Bachman is an associate professor in the Chemistry and Biogeochemistry Department at Cal Poly San Luis Obispo, and the associate director for the Center for Coastal Marine Sciences. I do not know that.
Oh, yeah.
Look at you. She earned a BA in chemistry from Pomona College and a PhD from Scripps Institution of Oceanography. She's a member of the newly formed Chumash Heritage National Marine Sanctuary Advisory Council, is on the executive committee of the Morro Bay National Estuary Program, and is a member of the Southern California Coastal Ocean Observing System, or SCOOS, Board of Governors. I'm very excited because her research interests center around seawater carbonate chemistry, something my lab group back there cares deeply about. And yeah, she's going to be sharing with us today some of her work to improve the characterization of local seawater chemistry along the California coast. And with that, I'll turn it over. All right, let's give a round of applause.
Thanks, Alyssa. Thanks, everyone. It's good to be here. I was chatting with people throughout the day. I think it's been at least 10, maybe 15 years since I was here as a graduate student. So it's fun to be back. I'm going to take you on a deep dive today of Morro Bay Estuary, which I guess I'll talk about more in a second, but has become very near and dear to my heart. Starting first with my funders so that I don't run out of time at the end. A lot of the work that I do is funded by SunCoos. And I've had an amazing partnership with the Morro Bay National Estuary Program from the very beginning. They have supported me throughout the years. I've also worked really closely throughout, I guess, my career at Cal Poly with Dr. Ryan Walter, who's a physical oceanographer, and a handful of other people in Morro Bay. And so, I mean, as many people say, right, it takes a village. And the work that I'm going to talk about today honestly was mostly completed by my undergraduate students at Cal Poly, right? A unique thing about Cal Poly is that we don't have PhD students, right? And so the research that we do is almost exclusively undergrad driven. At lunch, we were talking about my little army of undergrads. And I'll highlight some of them here and there who really made all of this work happen. A splattering of master's students as well. So sort of the two main questions that I made this seminar around, or like, I guess the story I'm going to tell today, are what are the drivers and impacts of the observed variability that we see in Morro Bay chemistry? So what is that observed variability? I'll talk about that first, and then think about what those causes are. And then really, why am I asking those questions, right? Are thinking about how well can we autonomously monitor chemistry in Morro Bay? How effectively or how usefully, right? Like effectively meaning both accurately and also something that's useful to stakeholders. And I didn't include it as one of my questions, but I'm thinking a lot about carbon cycling and carbon export and whether these sorts of estuaries sort of are important in that space or not. So what I'm going to run through today, introduction to Morro Bay, some of our early observations, freshwater contributions that turned out to be quite important, and then that idea of alkalinity estimation and carbon export, and then I'll bring us back with a couple little anecdotal oysters and seagrass stories. I know we have many seagrass enthusiasts in the audience here. So just to get to it. So Morro Bay Estuary, almost 300 miles south, I guess a little bit southeast of here. So Cal Poly is located exactly between San Francisco and LA, pretty much. And Morro Bay is about 15 minutes from campus. I spend a lot of time there thinking about this estuary. It has some analogy, it feels like to me, having been here for 24 hours now, to sort of Bodega Harbor, in that it's a relatively small estuary, right? It's about 6 kilometers in length. It's actually a bit human modified. The mouth of the estuary used to be at the top here, and this area was filled in. This is Morro Rock at the north end, and it was like actually partly blasted, and the rocks were dumped to close off this entrance to make a safer, calmer harbor, right? And so then the mouth was put this way with the jetties, calming waters and things. This is Morro Bay community, which is like a very coastal, small town, water-focused environment. At the south end, we have, wrong way. At the south end, we have Los Osos, which is, another small town. Both of these are like very, very linked to the estuary. The people who live here love this environment. And it's almost every time we are out in the estuary working, there's someone who's coming to ask us what we're doing, or if we're saving the eelgrass, or like what it is that we're working on. And that's something that I really like about it, right? I really like working and thinking about a space that people really care about. Okay, so what else about Morro Estuary? It's extremely shallow. So I'm giving you bathymetry colors here, right? Where red means higher, right? Less depth, and blue is deeper. So we have this channel that runs from front to back. The channel is dredged at the mouth every single year, and it's dredged further back into the channel every handful or so of years to keep that path free. Because there is a lot of commercial fishing, recreational fishing. There's actually a lot of people who live on boats throughout the estuary, like particularly in this region or throughout the bay. And then everything we see here in red and yellow is essentially mudflats. And I'll show you in a second, is variably covered with different ecosystems. We do have a bit of a salt marsh up here that very, very rarely floods. Like king tides only does it really get covered with water. A fun and complicated thing about Morro Bay is that the back of the bay, what I'll call the back of the bay, and when I really mean this area back here, is not the head of the bay, right? The fresh water that comes into Morro Bay when it does come in, right? Morro Bay is a low inflow estuary. I can never talk about them without thinking of John. But is Chorro Creek, which comes sort of through this channel here, and then Los Osos Creek, which comes through this channel here. So we kind of have this situation where the fresh water is coming in almost mid-bay, right? And then as the tides come in and out of Morro Bay, we see the water come back and forth. So it kind of makes for a little bit of interesting mixing dynamics where we can't, I still sort of will use this model of like an ocean end member, right? That's why I put up this sort of plot here, thinking about ocean dynamics or upwelling along our coast, right? That's water that is the source water to Morro Bay. It's coming in and out of the bay every tidal cycle. But then we also have this like freshwater component, right? Where we have intermittent rain. Some years we have a lot, some years we have none, and it comes out through our creeks, sort of in the middle. I have been working, I guess, to untangle those two parts. We have lots of otters in the bay, we have lots of seals, we have lots of sea lions in the estuary, and we have lots of eelgrass. Oh, something I don't want to forget. So I mentioned the Morro Bay National Estuary Program. I also have been lucky enough to work with some community scientists, people who live in Los Osos and who care massively about climate change and who want to make sure that this estuary is healthy and it really is not being acidified. That was sort of their initial concern. Their group is called Homefront Environmental Justice, and they have been really wonderful collaborators who meet up with my students and do surveys of atmospheric CO2. And so we have this kind of relationship between they measure CO2 in the atmosphere and we measure CO2 in the water, and then we can do comparisons or think about the variability in those two systems. That has been really fun. We also do some education and outreach work with them to the local high schools. Okay, so I mentioned seagrasses, and this is really what initially pulled me in to this environment. I got to Cal Poly in 2017 and met with, at the time, Jennifer O'Leary was our Sea Grant Extension specialist, and she was working with the Morro Bay National Estuary Program to think about this eelgrass, I guess I would call it a problem, right? Historically, the community, everyone identified as Morro Bay as being an eelgrass environment. There's like work that was done decades ago on the eelgrass populations in Morro Bay, about transects, about replantings, all these things. And when I got to Morro Bay, there was not eelgrass, right? We were down to estimates of 13 acres throughout the estuaries. And you can see it almost was exclusively, we call this Coleman Beach up here, concentrated in these sorts of edges of the deep channels. And the Back Bay was just mud, like just mud, nothing but mud. And pretty much everyone was horrified and very curious and confused and really wanted to understand what had caused this. And I think they hoped that I would, be their savior, figuring out the answer, which of course was naive, but at least that maybe I could give them some clues or help them understand what really might be happening. And I took that as an opportunity to really think of if we can characterize what the chemistry of this environment looks like when it looks like this, right? When it looks like mud, when there's very little of a photosynthetic community, then maybe I would have this like beautiful after and before story where maybe the eelgrass would come back. And wow, did it. And what this doesn't show is that actually pretty quickly after 2017, the eelgrass population started to recover, and it has been quite high for many years now. And so much so that in 2023, 750 acres, the estimate, we use drone surveys mostly to estimate this, were arguably the largest or the densest population that we've ever seen in Morro Bay. So in the time that I've been working in this estuary, we've had sort of these two extremes. And that to me drives a lot of really interesting questions, right? About what we see or like what those differences might look like in a chemical signature, never mind a biological signature. Okay. So seagrass population and loss or return of it was a big motivating factor for my work, but then so were the oyster farms. We have two commercial oyster grow-outs in Morro Bay. Grassy Bay Oyster Company is on this mud flat here, and Morro Bay Oyster Company is tucked actually closer to the Los Osos Valley freshwater input, which is interesting. And these oyster companies are a part of our community. I mean, I think you all are familiar with that here, right? You have local oyster companies. I formed a relationship with George, who at the time owned Grassy Bar Oyster Company. He was a biologist, actually, turned oyster farmer. He's since sold the business to a colleague of his and holds fast. So we have some new business owners, new stakeholders in our area who are also scientists and very interested in thinking about oyster health and ecosystem health and things like this. So they're actually really great partners. And yeah, what else about oysters? So when I first started, Grassy Bar was doing a lot of on-bottom oyster cultivation. So like oysters in bags laying on the bottom sediments. And through some casual conversations with George, we talked about how the oysters actually weren't doing that well sitting on the sediments. That maybe they weren't calcifying, they weren't calcifying as hard as you would need to take an oyster to market. And so he was having to move oyster, and I think they still do this to some extent, move oysters from the on-bottom bags to some version of like a hanging or tumble environment before they could sell them, right? So like it's an extra step in their already somewhat vulnerable business practices. So we had some interesting conversations about acidification and like if that was really a threat for them or what that looks like, and how you balance threats of acidification in amongst the other threats that they face daily, right? That the warming in the Back Bay, turbidity is a big contributor, right? The sand moves around a lot in Morro Bay, and we think that actually might play a big role in this entire story in eelgrass and in the oyster story. So I have worked with them throughout the years. Now at Cal Poly, we have Kevin Johnson, who did his work at Santa Barbara, and he's our Sea Grant Extension specialist there who works very closely with the oyster farmers to try to essentially learn as much as we can, optimize their work. Okay, and so then my last bit of introduction to Morro Bay is more about the physical environment, right? So Senkus we have 2 Senku shore stations that have been in operation since 2007. Ryan Walter was running them before I got there. We now are co-PIs on that project. And we have a sensor platform at the mouth of the bay, at the T pier, if you're familiar with the area. It's down below the Coast Guard platform, and the instruments sit on sort of this metal plate that we can lower down on a pulley and pull back up for maintenance, which makes it easy-ish to maintain. In the back bay, we have this pole stuck into a tripod in the mud and has a solar panel on it for the weather station, and then has sensors that are sort of attached to the tripod. We have trouble with sedimentation back here. We're actually looking into converting this into a buoy situation so that we won't have the same sort of sedimentation. Also changes your data a little bit because it'll be top anchored instead of bottom anchored, right? So tides are a little bit different. But at these stations, we get temperature, salinity, chlorophyll, turbidity, DO, and pH in near real time, every 10 minutes. And I think you all are mostly familiar, that data is available to the public in real time. So what does the physical environment look like? This also gives you an idea of like the mud flat nature of it. It's like very, very shallow and muddy. And even those channels that were drawn so perfectly in that bathymetric map, they migrate around a little bit and come and go depending. This image was actually the first time I had quite realized just how much Morro Bay Oyster Company was sitting like in this freshwater. It's not really freshwater because it only runs here, but in this channel of fresher water. And then you can see all of their oyster lines up on the mud. So here's just 2 example years of the physical environment we see in Morro Bay. So now on the map, you can see this, we call it the Bay Mouth Station, and then we call it the Back Bay or Bay South Station. And Bay South is in red, the mouth is in black. And so temperature, we see like a typical, as expected, annual cycle in temperature. It's warmer in the summer. It, in particular, is warmer in the Back Bay, which makes a lot of sense if you think about the fact that most of this is a meter at most deep, right? Lots of surface area to volume. Hotter in the summers, colder in the winters. Somewhat of a salinity cycle as well, although, I mean, Pacific Ocean coastal, about what we'd expect, 33.5, with the expected like low deviations in these sorts of rainy months. This is 2020 and 2021. We had pretty low, moderate low rain in those 2 years, but enough to see salinity drop a little bit. But we're still far above 30. These were relatively dry years for us. I'm not going to show you chlorophyll and turbidity plots that look almost like this, right? The turbidity and chlorophyll are both also seasonal. We have higher turbidity in the Back Bay. We tend to have larger chlorophyll blooms in the Front Bay, and then sometimes we see sort of a fall uptick in chlorophyll concentration as well. I think it's a surprise to no one, the oxygen and pH story is more complicated. Those sensors are more difficult to maintain. They're more variable parameters, and they're just more dynamic, right? There's more things that influence, right? Now we have biology at play instead of just the physics. We still see annual cycles, and in both dissolved oxygen and pH, but this time inverted, right? Low oxygen and low pH in these sorts of spring into summer months. We could think about maybe there being a contribution from upwelling at the mouth in those spring upwelling. We do see upwelling usually around April and May in this area. And if you squint really hard, maybe we see that in the spring, the bay mouth has lower pH, but then in the summer, the back bay has lower pH. This is only 2 years of data. I'm working with a statistician, Trevor Ruiz, and 2 of his students to work up the rest of this time series, right? We now have 2020 through 2025. And so I am excited to remake these plots and see how robust the signal is. And particularly the differences between the front bay and the back bay is sort of work we have in progress. I decided not to talk about it today. There's only so many things you can cover, but we see also some interesting coupling and decoupling in dissolved oxygen and pH, right? We expect coupling in pH and oxygen when there's a lot of photosynthesis and respiration, and there's a variety of things that can decouple those, and I think it's fun and interesting when they are decoupled. So we're looking more into that right now. Other thing I wanted to mention though, this is not extremely low pH, right? 7.7 is maybe low, and every once in a while, the upwelling signal we can see doesn't seem like it's represented well here, but every once in a while we see below 7.5 at the front bay at high tide, but we're not talking about a really acidic environment in Morro Bay, and that will come back in a couple minutes. So, familiar to many, if not most of you, but I actually use this image. Because I work so closely with the National Estuary Program, I give a lot of public talks where trying to explain alkalinity buffering to people is a real challenge. And I like this image as a model of how we might think about the carbonate chemistry, because the carbonate chemistry is not just about pH, right? Just knowing the pH of an environment does not tell you what the environment looks like, right? You can have pH 8 water that is fresh water, and you can have pH 8 water that is seawater, and those are very different, right? But you could also have seawater pH 8 water that is low DIC and pH C8, and seawater pH 8 water that's high DIC, and those are very different environments. And so I use this sort of like teeter-totter. I also have a 4-year-old, so maybe that's part of the teeter-totter analogy. To think about the fact that pH is sort of this balance, right? The balance between how much CO2 we have in a system and how much buffer we have, right? Which is really, the buffer is made of CO2, but how much buffer we have in the system, minus our protons holding up our alkalinity on that end, to think about this seawater pH. And so what I then spent, most of the rest of this presentation is thinking about these parameters instead of just thinking about pH, and thinking about how these parameters can inform what pH can tell us and what we can learn about the environment from them. Okay, so the other thing, 2 other things I wanted to mention here was obviously many more things contribute to alkalinity than just these things I mentioned here, right? And especially in estuarine systems, we are thinking about organic alkalinity contributions, as well as nutrient, silica and phosphate contributions. So this is admittedly a simplified picture. And then the last thing I just wanted to mention is that I do think there's potential that studying estuaries that are this small, right, my teeny tiny spot in Central California, can be important, because although most of the ocean is open ocean, surface ocean, and then the continental shelves, and estuaries are this teeny tiny sliver, they are thought to contribute in a meaningful way to global CO2 cycling, right? This is from Wei Zheng Kai's paper now back in 2011, but estimating, and he put these lines in where he thought the real answer was, but of how much estuaries might be contributing as a CO2 flux. And when you dive into the literature about estuarine CO2 flux and contributions to the atmosphere, both through degassing and through lateral transport, it's all about large estuaries. And there's good reason for that, right? The Chesapeake Bay is massive and so much water flows, so that even if it's relatively small margins or small changes, it still has a really big impact. But what I'll show you is that we have some pretty extreme chemistry in Morro Bay that makes me wonder if, even though it's a very small estuary and a small contribution, a big difference, right, can then also have a big impact. It's kind of the opposite end of that spectrum. And so I've been thinking a lot about this lately in the context of Morrow, in the context of Tamales, maybe Elkhorn Slough. Like, we have these really interesting estuaries up and down the California coast that are a little bit overlooked in this space, like in this space of estuarine, riverine to estuarine chemistry, carbonate chemistry, and global CO2 flux. So I think there's interesting space there. All right, so what did we observe? The first thing, how am I doing? Not great. The first thing I did was some transects throughout the bay. I did these with Ryan Walter. These are my students, Sarah Bartoloni, who ultimately published this work, and Daniel Sanborn, who's now at NOAA. And we did 3 days of transects through the bay at capturing high and low tide. Let's capture the extremes of the day to see what the extremes in the chemistry in a single day are. And then our goal was to capture 3 months in the summer. Ideally, which we missed, was before and after a salinity inversion. The physical data looks something like this. At high tide, the channel is mostly ocean water, right? Is filled with ocean water. But then at low tide, and so what that means is that at high tide, what we're seeing is ocean water, but all of the water that used to be in the channel is now spread out over these mudflats, right? Being modified, interacting with the sediments, interacting with the atmosphere, interacting with the organisms. And so at low tide, the water flows into the channel and you capture that signal. We can't easily measure the chemistry out in the mudflats. Some of you have tried to walk out there. It's not easy to access. It's very shallow. Even my little whaler that I drive around Morro Bay will get stuck. And so while this study is set up as high tide and low tide, it's also kind of a study of like channel versus like the rest of the mudflats, right? And like front bay versus back bay. And so a challenge of working in estuaries is that you're playing with space and time all the time, right? And so in this, right, when I talk about these low tide values, what I'm kind of talking about is the chemistry of all of the mudflats. I just capture that chemistry at low tide. Our first main outcome from this was thinking about the flushing time in the bay. How long does water spend in our estuary? Which gives you an idea of like how long it has contact with these modifying processes. And we found that in the summer months, right, when we can think of this estuary as a closed system, when we can think of it as having no freshwater input, which is maybe a reasonable estimate, we have a flushing time on the order of a week for most of the mid-bay, but up to 2 weeks, even a little bit more in the back bay. So water is spending a decent amount of time in this estuary. It's not like it's emptying and refilling every single day. There's a sloshing back and forth effect, which I think is pretty important and will become pretty obvious later. So this was summer 2018. We gather these transect samples, bottle samples back to the lab, total alkalinity and DIC values. At high tide, which is in black, we see sort of boring, flat-ish, kind of matches that temperature and salinity picture of relatively well-mixed water throughout the estuary. At low tide, we immediately see these increasing values in total alkalinity. And I think these were relatively modest. I was actually new to estuaries when I was doing this work in 2018. I mostly did, lab work or open ocean work in graduate school. And so I was excited to see these increases, but not blown away by them. 40 units is very measurable and meaningful, but not huge. And in total alkalinity, we saw almost no change in the pattern through the summer, which was an interesting observation. In DIC space, we saw something very different, right? So instead, we saw actually kind of matching high and low tide environments or concentrations. And then also different between our early and mid-summer months. To me, this indicates that there's more active control of the DIC environment, I assume by biological communities, but without more data, we can't say why. And notably, different ocean end member points, right? 2050 versus 2100 if we're looking at the mouth of the bay and going back into the back. So this point up here is all the way on the left. And then our late summer, we saw something entirely different, right? Where we just saw an entirely different environment in terms of DIC and therefore probably pH in the at high tide and then at low tide, where we saw these elevated DIC values, depressed pH at low tide. So that assumes some sort of respiration, right? Production of CO2 on these mud flats. The last thing, or well, not the last thing, the next thing we did with this work was think about the relationship with salinity, because our idea was, okay, we're seeing these hypersaline waters, we're getting high salinity in the Back Bay. How much of that is just what's causing the high total alkalinity? Like, total alkalinity is a conservative parameter. If you evaporate off water, you'll increase total alkalinity. Like, that's probably what's causing this. And this line here is where the data should fall if evaporation is what is increasing total alkalinity. And so, we calculated less than 20% of the total alkalinity change could be attributed to changes in salinity. So that's not it. I mean, there's some contribution by these physical changes, but mostly this has to be something else that's causing these high total alkalinity values and high DIC. DIC obviously complicated by the fact that gas exchange can alter this. So a little bit more difficult to track. The next thing we did was think about this in this sort of DIC versus total alkalinity space. This is a pretty common way to think about carbonate chemistry, although I think it is a complicated way and a way that you have to be a little bit careful. What we did was essentially normalized everything to that bay mouth value. So if we say that the source water is the ocean water that's coming in and look at everything as a change from there, then can we identify what processes are really changing things in our estuary? And I think at a broad scale, we can learn things from this plot, which is that we obviously have an increase in DIC, which we already knew from looking at our initial data. But we also clearly have a lot of other things going on. And those include things that could be dissolution, sulfate reduction, denitrification, processes that would be happening in the sediments. But what we also know is that there's a ton of calcification happening in the bay. I told you that there are oysters growing in the bay, right? They are calcifying, they are removing alkalinity. And so places where I think, and obviously photosynthesis is happening in the bay as well, although there was very, very little eelgrass at the time, there's still algae in the bay, right? There's still phytoplankton that are photosynthesizing. These plots are dangerous because it's really vector addition that you have to do, right? Many factors come together to create the final signal that you see. And so you can't just say, oh, like during, what was this, mid-low, we're nearly following the dissolution line. Obviously, everything is dissolving and causing this signal, right? We just really can't save that from these plots. But I still think that they can be helpful, I don't know, in giving us a place to work on. It started to make me think a lot about denitrification, and which is also known to be co-located with oysters. And I know Alyssa is excited to talk about sulfate reduction. So in 2021, we repeated this transect, partly to understand, is there any sort of temporal variability in what we're seeing? Like if we do summer again, do we see the same summer things again? And to capture a winter cycle, right? Because low inflow estuaries are wet in the wet season, right? If we have rain, then we have fresh water. And so we redid the summer and it looked much the same, although actually we had a bigger difference in DIC this summer, like a bigger gradient. And then in the winter, things were even more extreme in terms of alkalinity. The DIC signal looked about the same. We actually saw this really interesting pattern at the very Back Bay, which I haven't deciphered yet, I guess I would say. I could hypothesize about freshwater springs or things like that maybe. But we saw this very large increase in total alkalinity. And even more interestingly was if you plot the total alkalinity versus salinity, we see the summer samples line up in this way that you might expect. I think I have it on the next slide. And we see this huge deviation at the winter low tide, right? As salinity decreases in our estuary, we see an increase in alkalinity, and a substantial one, a really significant one. And that's really different than the open ocean. That's really different than most coastal oceans. So I grew up on this Malero 1998 paper, right, that gave us a formula for calculating total alkalinity from salinity in the open ocean. And it was based on this regression, essentially, right? This nice, beautiful relationship, total alkalinity increases as salinity increases. As we come back to this idea of alkalinity as a conservative parameter. Zhang did some really nice work in 2014, looking at some different environments and noticing that they are slightly different. But even what he calls coastal ocean was more scattered and random, but not inverted, right? Not the opposite slope. Ray Najjar has done some work in the Chesapeake Bay where they do start to see high alkalinity at 0 salinity. So once salinity gets very, very, very low, they see higher alkalinity. But even down at salinity 5, they're still seeing this positive slope in the alkalinity-salinity relationship. Oh, Ruben, I pulled this. I forget what the project was. It was a community science project where they had community members all up and down the East Coast take total alkalinity samples and send them in like at the same moment. They got this snapshot of coastal alkalinity. It's a very cool data set. And they have one data set that kind of maybe has increasing total alkalinity, but we're still in what I'm now gonna call very low alkalinity space, normal ocean space between 2000 and 2500. And so if I go back, this sort of inverted relationship where we have lower salinity having higher alkalinity is somewhat unique to the published literature, at least. Okay, in 1 second, nope, I'm actually just gonna, we did also collect nutrient data. I get this question a lot when I talk about estuaries because people worry about eutrophication. We do have nutrient data. The NEP has funded the nutrient data for a long time, although I'm actually now running it in-house. And generally we see elevated phosphates and elevated silica, but I'll come back to that in a minute. I want to keep moving. I have too much to say. So high DIC and TA during low tides, variable pH relationship with season. I didn't show you that data, but it is true. Slight salinity inversion in the summer, and then a full departure from expectations in the winter. I don't know if it's real expectations, but a full departure from the original model, and then elevated nutrients. We did start a program. My master's student, Jolie Higgins, started this shoreline Morro Bay monitoring program. We can't get out in boats all the time, right? My undergrads are not certified to drive the boats most often, and so we found the places along the shore that we could collect water. This means that most of this data is limited to high-ish tide, because at low tide, you can't get out on the mudflats to sample, so it's certainly biased towards sort of the less extreme version, right? It's biased towards the high tide data. Longer time series shows like what I called those relatively dry months. 2019 was a little bit wetter, and 2023, we had insane storms in the Central Coast. These are just our physical parameters. We see that daily cycle. We see the expected salinity cycle as we do our sampling. Total alkalinity and DIC confirm essentially what we already saw. At the front bay, throughout the year, we see some variability, a bit more in the early months and lower in the later months of the year. If we go into DIC space, the variability is a little bit more. You get photosynthesis and respiration cycles. In the mid bay and the back bay looks really, really similar. So I just am showing one as an example. We see much more variability within months, right? So interannual variability, and then much larger changes between the wet season and the dry season, right? And on this plot here, like my max alkalinity is 2,800. So we're talking like, I would say pretty high alkalinity for the Pacific Ocean, yeah? DIC, a little bit more of a confused pattern. Again, biological cycling having maybe a larger impact on that. This actually just visualizes the same data just in a different way in a heat map that my student Eric Smith has created. And it just really, what I find these plots really emphasize is how much every year is really different, right? And so this was the first driver that like the freshwater is really what's driving the variability in this bay. Because if we don't have a lot of freshwater, 2020 was a rough year. If we don't have a lot of freshwater, we see, you know, still variability, but much smaller, but then it just gets entirely swamped by having a big rainy year. So this was sort of our first idea that freshwater was playing a really big role in estuarine chemistry and then in estuarine carbon cycling. We also have been collecting nutrient samples throughout this time series, this shoreline time series, and this is phosphate, sorry, the labels are too small, showing relatively high-ish, I mean, this is 1.1, so not high phosphate, but relatively sustained phosphate, not really drawn down phosphate throughout both the bay, front to back bay, and also throughout the year. Lowest, it seems, in the late winter. And then nitrate has this really interesting pattern, which is the front and the back bay in green and blue have higher values in a pretty big intraannual cycle. And the mid bay, which if you remember is sort of where the freshwater comes in, we actually have, and where those oyster farms are, we actually have this drawdown in nitrate, right? So the N/P patterns are interesting throughout the years and not very predictable. And definitely not if we went back to like an oceanic model of what you might expect a 16-to-1 ratio to be. We're nothing near that, right? But I don't think that is too surprising. I also did mention high silica, and yeah, it's not really silicate, high silica, and we do see in the back bay high silica. Again, to me, this indicates contributions of freshwater, contributions of rock dissolution. So we have this seasonal variability in addition to tidal variability, in addition to spatial variability, of course we do in the bay, but it's really strongly driven by this wet cycle. Again, I didn't show you this idea that seawater pH is relatively stable, but I don't know, I need to look at the autonomous data. The discrete data is difficult to interpret within pH. It seems, oh, sorry, I can't touch that. It seems, it seems variable. Interesting N/P ratios. And then we did do some of these atmospheric CO2 comparisons that I mentioned with our community partners. And again, not as a surprise, but as a confirmation, the estuary is outgassing, right? The PCO2 of seawater, if we calculate it from some of these parameters, is greater almost always than the measured CO2 of the atmosphere. And so we are seeing that CO2 flux out of the atmosphere, even though I didn't show you that data. Oh, I keep forgetting my clock is wrong. Okay. So then we spent a year looking at the freshwater. And I said we had Chorro Creek and then Los Osos Creek going into the bay. And my student India led this project and went out and measured whenever we could. We tried monthly to get these different locations to characterize these two systems. And here's what she found. Nitrate is here on the left now, apologies. And if we look at Los Osos, so Los Osos upper is this green here, is sort of low, exceptionally low nitrate. But once we get to Los Osos lower, we see huge nitrate concentrations. This is a compilation of data over a couple years, but often the data is sort of like in the 40s and then higher as well. I mean, I guess these are violent plots, so you can see it. Not a huge phosphate signal. Granted, it is phosphate 2.5 in that lower region in the blue dot, so that is an elevated phosphate relative to a surface ocean. But not insanely high. We think of Los Osos as being sort of agriculturally driven, as this probably being fertilizer runoff into the freshwater. Chorro Creek is a different story. So if we start up at Dairy Creek, this is like our relatively clean water, almost no nitrate, almost no phosphate. And if we move lower into mid and lower Chorro, we see large phosphate concentrations, like very large, 15 micromolar, and no nitrate. And this is likely caused by the fact that we have a wastewater treatment plant that outflows into Chorro. It actually has a mandated discharge into Chorro Creek, and so it's supposed to flow Chorro is supposed to flow to Morro Bay year round. It doesn't actually, the water doesn't quite make it, it's too dry. But I think this signature and the signature of phosphate that we're seeing in Morro is likely a result of the fact that wastewater treatment treats for nitrogen, but they almost never treat for phosphate. Right? Nitrogen is considered the pollutant. And so what we have is this sort of like really interesting two creeks and two stories that are happening. What both creeks have is insanely high carbon. So our measurements of total alkalinity and DIC in those creeks, both of them, range up to 8,000 micromolar. This is pretty much the highest I can find in the literature. These are fully freshwater samples. All samples are salinity 0. They're all in the creeks, and they just have a ton of bicarbonate, presumably, a ton of carbonate and bicarbonate. There was sort of interesting trends within when the rain happens and when the values went really, really high. Although this is all surface water, I assume that there is some contribution of groundwater in order to get values this high, because I just don't see how water running over rocks would collect carbonates this quickly. I see, yeah, great, I love it. And just to give a comparison, I saw this presentation at SURF last fall. Daw gave this is work out of Princeton, which compiled globally riverine input and calculated and measured DIC and TA, and they excluded any small estuaries, of course. But the max they found was 4,000, right? So still high, but not so high. And those max values, I think the triangles, were all from like terrestrial forest areas. Like, so very different landscape than what we're seeing. And so this is where I really started thinking about how, yes, maybe Morrow is small, but it also might be mighty. Like we're having these really big, like very large carbon inputs into it. And then my next question was, where then is the carbon going? Obviously, if we have alkalinity and DIC with salinity 0 water coming in to the bay, there must be a different relationship between alkalinity and salinity. This data was all taken essentially from the mouth of the bay where Chorro hits Morro Bay. And across the full salinity scale over the year, right? So when the creek is running high, we get salinity 0. In the summer months, we get up to 35. We see this actually pretty nice regression between alkalinity and salinity that is exactly the opposite of what I had been told my whole life to expect, right? And then even at that point, right where the estuary hits, we're getting these 7,000 values. So those are entering the estuary. Like those are going right next to Morro Bay Oyster Company. They are crossing the seabeds. If we expand to more of the data, this is the shoreline data, the front and the mid-bay, we just see this scatter shot, right? Like drawing a regression line through this just doesn't even feel fair. At the mouth of the bay, probably we see this sort of relationship, but then we just see scatter later. So my Frost student, Emma, saw this and decided she wanted to figure out if it was leaving the estuary. And so for the past year, she's been going out to the Front Bay site every single week on Friday, or sometimes sending out people to take samples for her, and gathering water at 3 different depths. So these plots are over time, they're interpolated. They're interpolated in space, right? But then they're also interpolated in time because it's a single point each week that has been smoothed together. So important to keep that in mind. And she's used seawater pressure here to indicate high and low tide. And once the rains came in January, the fall was pretty boring. Once the rains came in January, it took about a week. And then at the low tides, we started seeing this signal show up at the Front Bay. So like that water, 1 week later, alkalinity, whatever, 6,000, 8,000, is coming, working its way through the estuary, and we're seeing it at the near Front Bay, not all the way outside, right, but right near the front. More rain in February, and we get these more extreme values. Granted, we are now back in the 2000s, right? We're talking more extreme, like we're getting up towards 2,300, which is high for Morro Bay, but not as insane. I guess this one's DIC. TA up in the 2400s. So we see carbon getting to the Front Bay, and surely this means that it's messing up our TA salinity relationship there. And it definitely is. So measured total alkalinity and salinity, much of the year looks something like this. And then when it rains, January, February, we get these departures. Interestingly, not repeatable departures, which is important for a moment later, but these departures. No longer a really clean regression, but still the same direction. If from salinity, we were to calculate total alkalinity, for this, she used Andrea Fassbender's work out of coastal Washington. It was the most coastal data set that we could find. It still has a positive alkalinity to total salinity relationship, but it was the best we could find. If we calculate it again, we get a reasonable estimate for much of the year, but during those rainy seasons, we get quite poor estimates of alkalinity, right? Wrong by 200, right? A 10% reduction. And that, as a Senkus PI who measures pH autonomously, starts to mean a lot for me. Okay, I need to finish. Sorry, I went too long. So we have high nitrate and high phosphate from our 2 creeks, very high freshwater, and then our estuarine high carbon is not constrained to the back bay. Why do we care about this? We have oysters in the back bay. One of the main reasons we do this sort of Senkus monitoring is to work with these stakeholders and inform them and try to support them. So we have what's called the Oyster dashboard, which breaks down the Senkus data into what is hopefully a more digestible format that an oyster farmer might actually pull up on their phone when they're worried about something. I have an oyster dying, like, is it super low oxygen? Is the temperature high? Like, what's going on? And what is the pH? I want to know, so if maybe what an oyster farmer really cares about, rather than just pH, is the omega, the saturation state, right? The equilibrium environment that their organism lives in, whether they are likely to be able to calcify or not. We know that organisms calcify better or more easily or less energetically at higher omegas. This is omega calcite. You also could calculate omega aragonite. It just looks the same, just down. And if we take sort of like our Senkus pH data, our findings about TA and salinity not being well related, and then calculate omega. So we compare a measured omega from autonomous pH and measured total alkalinity, and compare it to omega that's from a measured pH and an estimated alkalinity. We get this medium, small, large mismatch. So again, much of the year, it's a totally reasonable estimate. We can estimate alkalinity and pair that with a measured autonomous pH and get a reasonable estimate of omega. And then in the rainy months, I would say it's not a great estimate, but it's an underestimation, which I think I find more comforting than an overestimation, right? At least the estimation sort of would make someone worry more. I don't know if that's good or bad. But if you estimate omega from estimated alkalinity and measured pH, you underestimate the omega situation. And so you might cause, I don't know, worry to an oyster farmer if that's something that they were concerned about. So this was one of, this was like essentially Emma's like main finding from her work, that you are going to have to be really careful when you calculate omega from estimated total alkalinity. And this year that we did was not even a particularly rainy year, right? Like if we went back to 2023, I think that this picture would be much, much worse, and the estimates would be more terrible. I only have a couple minutes. I want to give some time to ask questions. I've also worked to deploy sensors at the oyster farms to try to understand a little bit more spatial heterogeneity. So we have sensor stations here at Bay South, and it's not far to the oyster farm, but these environments can be really spatially heterogeneous. And so we deployed an oyster pH sensor. Yui let me borrow one. And this data is really messy because oyster farms are really hard to measure. Like they go dry constantly. That's the point. They're intertidal. And pH sensors do not like to be dry and then wet and then dry again. And so all of this high data is light flooding. And I have a version where I filtered it out, but I kind of enjoy the cautionary tale. And all of this low data, I actually think was a very tiny pocket of water, was essentially the sensor going dry and then rehydrating. But right before and after that, there's some indication of like these very low pHs, well, lower pHs, 7.5, when low tide corresponds with night, right? And so again, the lesson that kind of came out with this was we're not seeing insane low pHs at the oyster farms, but we are seeing really important overlapping signals between day-night cycles and tides in space, right? And I mean, the takeaway there, right, is just that estuaries are complicated, right? It's hard to pin down all these different variables to say something really specific and to give a stakeholder an answer, like, is your environment safe? Depends on the day and the time and the particular year and how much fresh water happened. We've also done a cool comparison with Kevin Johnson and Christy Croker at Santa Cruz to compare dissolved oxygen at those two locations. So we put oxygen sensors out at the farm and then compared it to the Senku stations. And a little bit like that last slide, there is not a dire story here, which is fantastic, right? The sensors are doing a reasonable, not exact, right, but a reasonable estimate of what the environment is at the farms. And if anything, the farms actually see higher pH and higher oxygen than our Senku station. My assumption is that just because it's more shallow, so there's more gas exchange with the air, but I don't exactly have the reason. So the final story that I wanted to share with you all was to bring us back to seagrasses and this estuary that has undergone a huge change. And in 2021, Ryan Walter and I deployed some sensors inside and outside of an eelgrass bed. So this is an eelgrass bed right in the mid-bay, right here there's a harbor back here. And we put one sensor package in the channel and we put one right in the eelgrass. And we had some tilt meters to ask how different was the pH environment inside and outside an eelgrass bed, right? This was a time a lot of talk about submerged aquatic vegetation, pH amelioration, and even though we don't have super low pH in Morro Bay, right, we still want to think about this is going to be a huge change between a mudflat dominated estuary and a seagrass dominated estuary. And what we found was very surprising. So I included this top plot of temperature to give us this physical framework. And what you're seeing are the tides, right? The back bay, so the high tides are when the high temperatures are when low tide was flowing over the sensors, right? When we have that back bay water that's heated up through the course of the day, it was summer. And if we're in the channel, we just see this nice semi-diurnal sort of mixed signal, right? It almost looks like a tidal. But inside the eelgrass, we see a huge lag, right? And our interpretation of this, right, is that this is just seagrasses is an entire ecosystem, right? There is resistance to flow, and they're essentially capturing their water within the grasses and holding on to it. And it wasn't until the tide really started coming back in the other way, I guess I don't have my map here, but when the tide started coming back from the north, that it really flushed that warm water into that eelgrass bed. Happened a little bit less on the lower tide. But so then when we looked at pH, and this also was true with oxygen, was that when that happens, when the tide has flipped, but the eelgrass has sort of retained its water, that ecosystem, we're seeing these really low pH values, right? So that ecosystem is net respiration. Like that community that's created by that ecosystem seems to have some sort of a net respiration signal compared to the adjacent, very close mudflats. So these were very surprising results. But again, it's not surprising that eelgrasses are an entire ecosystem that have respiration. And one of our main takeaways, right, was that the physics is so important. How the water flows is so important, right? Whether that water is stagnant or changing, mixing or not, plays a huge role in whether or not you get extreme chemistry. Stop there. We have large seasonal changes in seawater conditions, freshwater contributions that I think have been underestimated for how they impact the bay, partly because we don't have eutrophication, so it's easy to write off, right? But we do have really high carbon, and I think that could be pretty important to the ecosystem. Different point sources of our nutrients, and this idea that maybe constraining carbonate systems with our autonomous system is a little trickier than we thought. And I'm super curious to see how other estuaries along the coast, if they're experiencing that or not. I was hopeful that I could just estimate alkalinity from salinity, right? And then with pH, I have my constrained system. But I just don't think that that's realistic. Definitely not in the back bay, but even in the front bay, I think it's just not possible. And then finally, this is my other example of why autonomous shore stations are difficult, right? Everything fouls, constant. This is like the little ISFET on the pH sensor. And then finally, just ending with this idea that it's been strong partnerships that have made this work possible, and it's strong partnerships that I think give this work meaning. And I don't know, I guess I wouldn't be here without them. So with that, thank you very much.
All right, thank you so much, Emily, for the awesome talk. I know we're rated 4, so if anyone has to sneak out, totally understandable. But for those that would like to stay and ask a few questions, does anybody have anything they would like to ask first? Don't be shy. Yeah, oh, okay, yes.
Great talk. I found it super interesting. I was just curious if you had like any interest in looking further into this question about groundwater, or maybe if not you specifically, but just like collaborating with other people nearby who do that kind of research.
Yeah, this question is actually even funnier because I'm married to a hydrologist who studies groundwater in the SLO region. Yes, I am interested. I think I have to be, right? Because now I can't not understand the answer here. It does seem like we have Monterey Shale in the area, which is very carbonate-rich, area, and so I think that's probably what is contributing. I worry less about groundwater contributions to the estuary directly, although I'd be interested to measure them, because we actually have problems with saltwater intrusion in Los Osos. Like their freshwater is being contaminated by saltwater because essentially they're pumping, right? And so historically Los Osos had a septic system. I think there's like a little bit of play in there, but they've since converted to sewer. But we actually see saltwater intrusion. So I'm not sure I expect there to be a lot of direct groundwater input into the estuary, but I am really interested to figure out what's the groundwater contribution to the freshwater that we're measuring and why that's happening sort of, or like it must be some function of recharge somewhere else and an expression. I think it's I'm really interested to find out. My colleague also, I co-advise my lab now with Dr. David Long, and he lives in Los Osos, and he swears that there's a spring that runs year round. So that's this year's plan, is like we will track it down and figure out what the chemistry of that spring is. Yeah.
Other questions? Yeah.
Thanks for your talk. I was amazed to see how much recovery of eelgrass there was between 2017 and 2023. Seems like a lot of times there's a lot of work to achieve a lot less restoration, but that apparently all happened naturally. So I'm wondering, well, you can answer, but I'm wondering what's known about causes of that recovery.
Yeah, something that I should have mentioned that I'm glad you remind me is that we, a royal we, the NEP, myself, and those who work in the area do think that the recovery was mostly natural. And I did, the NEP and Ryan and I did receive funding from the EPA through the CWG, the California Water Working Group, to do a little bit of eelgrass restoration. And so he and I, some of the data I showed you was funded as a part of that project. And what the NEP did was eelgrass replantings. So a bunch of different studies on what was the most effective and not effective, and a lot of monitoring. And so they did take shoots from healthy eelgrass populations and distribute them around the bay. They did not see obvious recovery. Like they did not see the 1 meter plots that they planted turn into 10 by 10, but they did then see nearby eelgrass. And so we don't know how much recruitment was related or unrelated. It was really hard to track. They did a very nice job tracking it. It's just very hard to interpret. So we did have efforts at recovery, like from a human intervention side, but I just really doubt that that is what caused it to go from 50 to 750. Nature has to be at play there. If I have to put like an answer on why I think it's probably having to do with elevation and sediments is my guess, is that there was some sort of turbidity event maybe that covered, that's our best guess, is that the bottom has been moving around, but we really don't have an answer.
Oh, yeah, we'll do one more. I think we have time.
Then kind of similarly with the oysters.
Yeah.
I was trying to synthesize all the data. So you were saying that the folks that are growing oysters, at least at some times, have had trouble getting a lot of calcification near the bottom.
Yeah, like a little bit of a chalky shell.
And then they've had better luck if they hang them. So given all the data that you've been collecting and seeing, do you have a hypothesis of like what's going on with the chemistry?
Yeah, I think two things. I think I have two ideas. And one is just that you got a whole bunch of oysters that are respiring a ton. Like once they're large, like that's a very large respiration signal that's lowering oxygen and pH. And then you put it in very, very, very shallow water, and it's easy for that water to get extreme, right? So the water is flushing off of the farm, but leaves a very small amount behind that's stuck up in the oysters, right? And so I think it could be very extreme chemistry within there. But then also they're lying on top of anoxic sediments that I think could be corrosive. And so which one of those it is, I don't know the answer. Yeah, but I do think that's probably why, and why they've switched to more of a hanging culture, which I think probably lets the waters, like they still go dry, but I think the water probably drains out in a more repeatable way. Yeah.
Let's measure it.
Yeah, let's measure it. Yeah, my last pitch is that I know a lot of you study seagrasses, and like we would encourage you, please come down. I know some of you already do and work in Morro Bay. We'd love to know more about the seagrass population. The NEP does really wonderful jobs. Their funding is very federally dependent, how much work they can do. But we really don't have someone at Cal Poly who's doing like plant ecosystem kind of work. There's a professor at Cuesta who's been doing some wasting disease questions with some undergrads. And we tried to sort of hire a marine botanist kind of carbon cycling person, and then instead we got a wonderful intertidal ecologist. So I don't know. It's only 300 miles. Yeah, come on down.
Awesome. Well, yeah, we will all try. I know I will. Well, let's thank Dr. Bachman for an excellent talk.
Thank you guys.
And thank you everyone for coming. Again, our seminar is picking back up, so this will be weekly Wednesdays at 3 o'clock for the next several weeks. Thanks, everybody.